Hello, this is Matt, Tiger, and Issa here. We are the Table E from 7B Stripes. This is our experimental report, and we're going to start off with our question and hypothesis. So how do different types of sugar affect the growth of bacteria was our question. And we're going to prove this, or our situation that we're going to prove this in is we're going to be taking samples from the water fountains by Davidsville and putting them on an agar plate. Then we will transfer them to the incubator and wait a certain period of time. Our hypothesis is that if you put glucose, that glucose will have the highest number of bacterial colonies because it is the simplest and easiest for bacteria to consume, to break down and consume. The next one would be none because technically it's just a plain agar plate with bacteria on it, and the next one would be both. No, sucrose. Sucrose is going to be a little harder for bacteria to consume because they also have to break down one layer, then another layer of sugar, and both will be our last and have the least amount of bacteria because it has both glucose and sucrose. The glucose doesn't really matter, but the sucrose will definitely put the difference in between in the bacteria colonies.
Hey guys, I'm Tiger. I'm in 7th grade. I'm in the stripe section, and I'm in a group with Matt and Issa. And here's my expected graph. And I did it for glucose, and we think that glucose will have the most bacteria colony aside from our positive control, which is both sugars. And we think it will have more bacteria colonies because it's easier to break down and for bacteria to grow off of. And then here's Matt's graph of sucrose. We think sucrose will be harder to break down, and so therefore it will take longer to make bacteria colonies. And then here's both, and it's just the two showing the two lines on one graph. And I did the combined report as well, and we each did our own independent bacteria report, but I just combined them and wrote a few sentences on why just to make the sentences flow together. And we added links from the bibliography, the combined bibliography. All of the sites in there are sites that we use to find research for our for background knowledge about bacteria. And so you can see there's websites in parentheses, like right here. And then also I'm filling in for Issa
because Issa is taking a vow of silence, so she can't really participate in the report.
Even though she's already said some words. But, okay. Issa's in 7th grade in the stripe section in a group with me and Matt and I. She wrote the materials and procedures for our document. The materials section tells us what we need to do in the what we need in the experiment. In the experiment, why we need those materials, and where we plan to get them. The things that we need for our experiment are plates and agar to grow bacteria on, Sharpies to label the plates, the liquid forms of 2 sugars to help grow the bacteria, parafilm to seal the plates, Q-tips to transfer bacteria onto the plate, an incubator to speed the growth of bacteria, and a water fountain to collect the bacteria from. She also did the procedure, and in the procedure, we just wrote down all the steps. It was actually pretty simple. There are also pictures, as you can see. This is Issa labeling the plates before pouring the agar into them. And then this is me pouring the agar into the plate. And I'm trying to avoid contamination by opening the lid just a little. Here's Issa and V, and I'm holding the Q-tip so they don't get contaminated while Issa swabs the plates. Here's Matt's hands collecting samples from the water fountain, and then here he is swabbing the plate from the samples that he got, and then here are the rest of the steps. And here's an example of a contamination, a plate that got contaminated. There's a bunch of mold growths around here and right there. And then here are our final graphs. This is the final graph for both. As you can see, the highest was 40, I think around 41 or 42. And the lowest was about, I'd say, 10. And we thought this was because, I think most of it was glucose because it's easier to break down. And then one reason that there might be a negative trend is because if the sugar solution is too concentrated, then the bacteria can't use it, and it'll upset them and throw off their balance. And here's the none graph, where the highest was around 31 or 32. The trend is also, as the number of the swabs rises, the number of bacteria colonies falls. And here's the one for glucose. The highest was around 31 or 32 again. And that is because the bacteria were able to grow off the sugar while they couldn't find in the none section. And here's the graph for sucrose. Now sucrose had a crazy outlier. It was around 87. We think this plate could have gotten contaminated for a few reasons. It could have the person inoculating the plates could have sneezed or coughed while they were inoculating the plates, and maybe we left it in the incubator too long. And here's the data table. And then, yeah, that's pretty much it. Thank you so much.